Thank you, Dr. Green. We'll take some questions from the audience. That's okay with you, from these two mics here, so we can hear you on the video that we're taping in the back. So if you mind lining up at the mics, that'd be great. Thanks. And we'll go back and forth.
Good evening, and thank you. That was, like my friend said, wow. As we're looking around the planet today and species are becoming extinct quite rapidly, some we're bringing back. Are we learning something from the bringing back of some species versus others versus what you've been describing about our history?
So as far as I'm aware, and my partner actually works on this, we haven't brought anything back yet, although people are working hard to figure out how to do this. But there are technical questions that are easier to answer and ethical questions that are harder to answer. And I wouldn't put money that anything will ever actually be brought back. If this is in relation to Neanderthals?
No, I was thinking of very small populations of species that are on the verge of extinction and then coming back, some very rapidly, and then we genetically study them, and some have greater diversity and survivability than others.
Yes, so are we learning anything about what makes things go extinct? Is that the question?
Kind of.
Kind of? Yeah. The answer to that question is a resounding no, sadly, from genetics. If there was a way to look at a genome and answer the question, will this organism go extinct, we would have done that for our own genome a long time ago. I mean, it's just not a reasonable thing to do now in genetics. What's interesting, what is often done in this field of conservation genetics is to go out, collect a lot of individuals, and ask how much genetic diversity is here. Because larger populations have more genetic diversity and smaller populations have less genetic diversity over time. But this can be extremely misleading, and I think is, in its application, is almost borders on dangerous sometimes. I think that there are well-meaning people who do this, but for example, all of the great ape populations are critically endangered. Chimpanzees, bonobos, orangutans especially, and gorillas. Humans, on the other hand, are the opposite of critically endangered. We're growing exponentially. There are 7 billion of us, and there are less than 100,000 or so of all of the other great apes. But if you look at genetic diversity, they all have more genetic diversity than we have. So if you were just to ask, how is this species doing based on the amount of genetic diversity, it would look like they're all fine and we're in trouble. We know, we don't need to use any complicated genetics to know what's going on. They're losing their habitat to us. And the signal of population size that exists in genomes, it takes a long time at a small population to reduce that diversity, and it takes a long time in a big population to recover that diversity. It's a lagging indicator, a very slow-moving lagging indicator of population health. But it's what we can do. It's what people can go and measure and do things like go and say, is this subpopulation different than this other one? And if so, we need to protect it. And the answer is always yes, it's different, and yes, we need to protect it. But it's not that complicated. It's habitat. Things are in trouble now because they're losing their habitat. If we protect their habitat, we protect the genetic diversity and we protect everything else. I think the genetics is almost even a distraction in this.
Thank you.
Yes?
Thank you for a really clear and concise presentation. This is a more technical question. Were you able to detect human accelerated mutation regions in Neanderthals and Denisovans?
Yes, yeah. So
any opinions?
There was an observation, a study done by a colleague of mine, Katie Pollard, and some other folks did very similar interesting things to when the human genome first came out and the chimpanzee genome first came out, scan across the human genome and find regions that are evolving extremely fast in humans that throughout mammalian evolution appear to be evolving very slow, possibly because their importance, and evolution won't tolerate changes in them, yet in humans seem to be evolving very fast. We did look at these in the Neanderthal. They don't appear to be evolving very fast recently or long ago in time. It appears that this fast evolution is equally spread before we split from Neanderthals and after we split from Neanderthals. So we couldn't really get a better temporal resolution on that. That entire kind of subfield of comparative genomics was turned on its head, I think, by this observation that recombination hotspots are largely driving these human accelerated regions, that where this recombination machinery that puts the two chromosomes together, exactly where that lands. I said to a first approximation it's random across the genome. Well, if you look in finer scale detail, there are particular places that if it's going to be in this neighborhood in the genome, it's going to be right here. And the recombination happens very fast there, and in the neighborhood, not quite as fast. Well, when that happens, if you happen to be heterozygous, have a different allele in mom than in dad, there's this phenomenon called biased gene conversion, where you're a little more likely to get either mom or dad's allele depending on who has the GC base pair. It's a little more thermodynamically stable, and it creates this bias in inheritance. So all of that's kind of weird, but in the end, the function of this is wherever these recombination hotspots are, and they change over evolutionary time, wherever they have landed, that region evolves very fast. And if you just look for the fastest evolving regions of the genome, you find where there are these historic recombination hotspots, which makes them less likely to be due to what we would imagine they were due to, some important change in humans. So they get to be less interesting from an evolutionary perspective and more interesting from a recombination perspective. But they're not particularly interesting with respect to Neanderthals, sadly.
Thank you.
Yes?
The Florian's bones are 30,000 years back from we are in an area of interest. So how do they compare with the Denisovan DNA?
I would love to know that, and I have a standing offer with Svante to go back to Germany as soon as he gets bones that you can get DNA out of. It's been tried, and that's an area of the world where DNA preservation is not favored. It's very hot and it's very wet, and they did try early on to extract DNA from those bones, and there was nothing recovered. But that would settle the mystery, if you could get DNA out of the bones.
Thank you.
Yes?
So now that you're studying the differences between the Neanderthal DNA and the human DNA very, very closely, and especially those areas that differ markedly, have you found any clues at all to the Neanderthal extinction from that study?
No, sadly, beyond the fact that they had a small population size for a long time, that Neanderthals, it was not a catastrophic event that killed them, or maybe it was, that finally did them in, but they were never a very large population that we know. It's also the case, and I didn't really talk about this, but the Neanderthals disappear from the fossil record in the same temporal and spatial pattern that humans show up in the fossil record across Europe, at least. They go away from east to west at about the same time that humans are on the scene and going from east to west. That does not, I wouldn't convict us based on that evidence, but I would investigate more for sure. It would be an amazing coincidence if we weren't kind of responsible for their demise in some way. I would guess, but that's not genetic evidence. Yes?
So in looking at these degrees of variation within Homo sapiens and I guess for primate evolution and evolution in general, is it still useful to think of it in terms of a species becoming another species and speciation? Or I noticed you actually didn't use that word your entire talk. And I'm wondering, is that just an outdated concept and we're now just looking at groups that have different degrees of biodiversity?
I love this question. Yeah, it's a, yes, to my thinking. There are many species concepts. You may have heard biological species concepts and morphological species concepts, different ways of classifying things into these groups that, and I am firmly on the soapbox here. There are other people, very smart people, smarter than me probably, who disagree a lot, but this is my view. This idea that things belong in these groups, this Linnaean idea that there are these types and everything belongs in a group. And each group, if you just look at it enough and classify it in the right way, the truth will be revealed how everything goes in each particular group. This idea, it breaks. It always breaks. There's not a concept of species that anyone has ever described that is not directly contradicted by some example that everyone would agree are different species. For example, a concept that we often use is if two groups are reproductively incompatible, if they can't have a hybrid offspring that's viable and fertile, they're different species. Well, we know that humans and Neanderthals could. Some people would clearly make those different species. We also know that grizzly bears and polar bears can. They do it in the wild all the time. They make hybrids that are viable and fertile and go back cross even more, and you get gene flow between polar bears and grizzly bears. If we're then going to stick with that as the species concept, how we define species, then there is no polar bear species, and we don't have to worry about the ice caps and just cancel all of that. But clearly they are different species, and that doesn't work. It doesn't work in that case. There are many, many cases like this where something called ring species, where there are a geographical distribution and neighbors across this distribution can hybridize and they're just fine across the entire distribution. But if you take individuals from the extremes of this distribution, they can't hybridize anymore. There are too many differences there. So where is the boundary in this continuum of where one species begins and the other one ends? It's impossible to say. And the problem is this concept. There is no thing like species. We have this because Linnaeus wanted to understand the mind of God and how he organized nature, and it just ain't that way. That we can often make hybrids between things, and things did hybridize. Things have mixed. They mix all the time. They're continuing to mix now. It happens in birds like crazy. It happens pretty much wherever we look, we see evidence of this. And yes, thank you for the question. All of this invented terminology, subspecies and races, and where exactly you draw the line. Why would we need that if there were these clear species and things clearly fell in boxes? It's all a symptom of the problem that is not a good description of how populations change through time.
Thank you very much.
Wow. Yes.
Back to Neanderthals, the red-headed gene that occurred in Neanderthals.
Sorry, the what gene?
The red hair.
Red hair gene, yes.
Yeah, the red hair gene, the deletion of certain parts of the chromosome. Could you elaborate on that?
Sure.
And did that happen?
Yeah, so
the frequency of this happening later on.
The variant that Neanderthals have in this gene is called MC1R that was published from Miki Hofreiter's group. We don't see that variant in the Vindia Neanderthal that we sequenced, but they did a very convincing job that this gene that has variants in it in humans today that cause red hair because of inactivation of this gene. It's also inactivated in a particular Neanderthal, this L-Cedron Neanderthal that they sequenced. So if the genetic background, red hair is not a single locus trait, but it requires a particular genetic background and then inactivation of this MC1R. If that other genetic background is the same, then the inactivation they observe would cause red hairs in Neanderthals. But we don't see that in any of the Neanderthals that we have sequenced. So if it existed, then it likely would have been, like red hair today, a rare thing.
Yeah, it's a gene complex, and
Yes.
it's not just one thing.
Yes, right, exactly. And that variant is also not a variant that's segregating in humans today, so humans did not get red hair from Neanderthals. It may have been the case that some humans and some Neanderthals have red hair, but we didn't get that gene directly from them.
Thank you.
This is kind of a general question. When you're comparing Neanderthals with us, there's variation among us. So how do you isolate what is the true definition of us and use that to compare it with Neanderthals? In other words, how do you take out the variability among us in order to make that comparison?
We don't, is the short answer. Every individual is a valid instance of us, and compare one by one each of us to the Neanderthal. So for the admixture test, this was a pairwise comparison of two individuals. It's not all West Africans against all Europeans. It's a particular West African against a particular European, and asking, does the Neanderthal match one of those more than the other? And then, of course, we can do lots and lots of individual West Africans and lots of individual French guys, but it's always a pairwise comparison of individuals.
So, for example, with your French guy, is that 100 French guys, or is it only one?
In that sample, it was just one. Since then, we have a larger sample. There's a project now called the 1,000 Genomes Project. There's publicly available complete genome sequence from more than 1,000 individuals from all over the globe. They all show more or less the same signal. But what's important to remember, and I think is exemplified nicely by that, that plot showing population size over time, one individual actually has many, many, many different independent observations of evolutionary history. Because of this process of recombination, each place in the genome is a different trajectory back through time, and sequencing an individual's genome gives you many, many hundreds of thousands of glimpses into the past.
Thank you.
The Neanderthal brain size was comparable. You said even on the average larger than ours. When will we know, or when will you find out, do you think, whether Neanderthals had speech?
Yeah, that's a great question. And if there were a good reason to bring Neanderthals back to clone them, that would be it, to try to teach them language. There's a very eminent anthropologist, Richard Klein, at Stanford, who makes a very convincing argument that one of the things that kind of sets us off as humans, this great leap forward, was the ability to communicate in a way that is fundamentally different than we ever could before, and sets us apart from Neanderthals. And there was, if you kind of look at the technology development through time, something happened about 100,000 years or so ago where we just hit warp speed. And maybe this was the full development of language. I think there's a convincing case to be made for that, which would, shut Neanderthals out, would put them on the other side. There is speech and language is compared to something like red hair, which is already genetically complex. It's extremely genetically complex. All of the genes that we know about, all one of them, FOXP2, that are directly involved in speech and language, look the same between humans and Neanderthals. But it's not, a very powerful thing. There are a lot of genes. We just don't know which ones they are to look for. But that, I'm rambling now. I would love to know whether Neanderthals could talk, and I don't.